Hello and welcome, everyone. My name is Emily Davis, and I'm a Senior Commercial Product Manager at IDT, focusing on NGS solutions. I'm thrilled to be your moderator for today's webinar on unlocking RNA insights in solid tumors and blood cancers using Archer FUSIONPlex panels paired with Archer analysis. Our guest speaker is Dr. Natalia Guseva from the University of Iowa Healthcare, and she'll be presenting on the lab's experience using Archer FUSIONPlex assays utilizing lung, thyroid, salivary gland tumors, and blood cancer samples to unlock RNA insights. I would like to thank the Association of Molecular Pathology for hosting this webinar, and we are excited to attend the annual AMP conference this November in Boston. We've slotted some time at the end of the talk for Q&A, so please submit your questions into the Q&A feature of Zoom. Before I bring in Dr. Guseva, I'd like to take a bit of time to introduce you to the Archer platform, so let's jump right in after our disclaimer. Archer products are for research use only. They are not for use in diagnostic procedures. The purchaser is solely responsible for all decisions regarding the use of these products and any associated regulatory or legal obligations. Archer offers a technology platform for next-generation sequencing library prep and data analysis in an easy-to-use workflow. The Archer platform is powered by proprietary anchored multiplex PCR chemistry and co-optimized algorithms run on our purpose-built secondary analysis software. These elements come together to enable users to identify a variety of mutation types, including gene fusions, splicing events, SNVs and indels, CNVs, gene expression, SMI, TMB, and HRD. We have product lines for RNA, DNA, and cfDNA inputs, and we have designed assays within each product line for multiple applications in the oncology and inherited disease space. Depending on your goals and available sample types, you can choose one of our four main product lines: FusionPlex, VariantPlex, LiquidPlex, or Muniverse, or you can build custom assays on our platform to fit your needs. Here I'm showing the biomarker coverage of our FusionPlex and VariantPlex assays. Starting with targeted RNA sequencing using FusionPlex not only streamlines your workflow, but also improves turnaround time by providing fusion, SNV indel, and RNA expression data upfront. Our parallel RNA and DNA sequencing assays are designed to scale and give labs the flexibility to design NGS workflows that suit their specific goals, whether that's starting with RNA or integrating DNA and RNA assays from the beginning for a more complete omic picture. Ultimately, this approach empowers customers to scale their biomarker profiling strategy with confidence and efficiency, broadening access to research insights. One of the key advantages of using assays built with AMP chemistry is its flexibility, which allows you to customize your assay and add new content without compromising performance. In fact, around 60% of our customers use custom panels. On this slide, we're showing 3 custom assays of different sizes: small, medium, and large. The large panel includes all the primers from small and medium panels. What's important here is that the assay performance remains consistent even as more biomarker content is added. This means you can start with an off-the-shelf assay or build a custom one, and it can evolve with your research over time. As new targets are discovered, you can expand your assay without impacting its core performance. Now, I'm very happy to introduce you to Dr. Natalia Guseva. Dr. Guseva is a Technical Director of Molecular Pathology Laboratory at University of Iowa Healthcare. She spent over a decade in cancer research before shifting her focus to the rapidly evolving field of molecular diagnostics. At University of Iowa Healthcare, she oversees all technical aspects of assay design, development, and validation. Her primary interest lies in advancing and interpreting next-generation sequencing tools for molecular oncology research. Please take it away, Dr. Guseva.
Thank you, Emily, for nice introduction. And I'm going to share our experience with FusionPlex technology here at the University of Iowa, and also some interesting findings from our research. So first, I would like to introduce the University of Iowa Healthcare. This is a public teaching hospital and the key part of the healthcare medical center. So we have close interaction between our teaching, research, and clinical care team. So the molecular pathology lab is involved in patient care, education, and research. And we perform mutation profiling for several types of cancer, including leukemia, melanoma, brain, lung, and some others. And we committed to develop new advanced molecular testing methods for oncology. And we use multiple NGS technique for both clinical and research application in the lab. Oops. So we have two primarily technique in the lab sequencing platform for Ion Torrent and Illumina, which support our comprehensive DNA and RNA sequencing. And now I would like to talk about our experience with FusionPlex in the lab. I'll share how we approach analytical validation of new assays, highlight few of our research studies, and touch on some of our published work using these assays. So we were early adopters of FusionPlex technology. We implemented first DNA sequencing about 15 years ago, but we had a need for fusion detection. The FusionPlex approach was very appealing for this ability to detect targeted fusions without prior knowledge of fusion partners. And also what was important for us, it had straightforward analysis without heavy investment in bioinformatics. We started with small assay for sarcoma fusions, and we gradually expanded it through thyroid, lung, salivary gland, and heme assays. And last year, we brought a comprehensive custom pan-cancer panel that integrates all previous assays and broadens gene coverage across solid tumors. So in this big panel include over 1,000 GSP gene-specific primers. So I will introduce our workflow. This is overview. Our workflow begins with total nucleic acid extraction from tumor samples, followed by FusionPlex library preparation, sequencing, and analysis using Archer provided software. The goal is to detect all possible fusions, oncogenic isoforms transcripts, as well as relevant single nucleotide variants and small insertions and deletions. Formalin-fixed paraffin-embedded tissues, FFPE samples, the most common specimens we work with. And we also frequently work with fine needle aspirations at small samples. They could be processed with FFPE cell blocks or prepared as direct smears. For hematological malignancy samples, we usually have either whole blood or bone marrow. So FFPE samples have known challenges, such as nucleic acid degradation or chemical modification, which we have to take to consideration. And for molecular testing of tumors, we have to apply tissue microdissection because you have to select specific region and also count nucleated cells with this tumor. Otherwise, this material could be non-adequate for interpretation. Some samples we have for testing is quite small, which can limit nucleic acid yield and affect assay performance. But we have been quite successful working with these samples, and one of these shown on this slide. This is an example where we had successful outcome from this small specimen. For RNA extraction from FFP samples, we use QIAGEN or an easy minikit. But we omit DNase treatment step and treat it as a TNase. And for blood, we use QIAGEN blood kit. This process automated for us. We use HiCube from QIAGEN. So a little bit about library preparation. FusionPlex library is based on uncored multiplexed PCR. and were targeted cDNA fragments selected through MBC adapter ligation and 2 rounds of PCR using gene-specific primer for targeted gene and universal primer for adapter sequence. This approach allows detection of known gene fusions and also some unknown partner for targeted gene. I also want to point out a little bit more about MBC adapters. They include short random sequences which called molecular barcode, and it's uniquely tag a regional nucleic acid molecule before PCR amplification. So these unique fragments and unique start sites together provide reliable measure of sample complexity, which can be correlated to assay sensitivity. This metric helps prevent against potential false positives called from PCR and sequencing errors. We had experience with sequencing this library on different sequencing machines. We use technology from Ion Torrent, and we use different Illumina instruments such as MySeq and NextSeq 550. And now we moved for a bigger, larger NextSeq 2000 instrument. And we do our analysis using Archer analysis software locally installed on locally installed virtual machine. So in our setup, sample demultiplexing occur on board of NextSeq, and the multiplex FAQ file automatically transferred to virtual machine. And we establish technical samples and sequencing QC parameters. So like for our analysis, we use fusion detection and variant detection. In software give you some specific QCs you can review at the end and make conclusion how it works. Usually it's if sample is really bad, it's flags that it's not usable. In the past, we used to run pre-seq QC for strand cDNA amplification because it's tell you about RNA integrity and predict sequencing success. But later we discontinued this step because our samples, like over 95% of all our samples, had a score below this threshold, and we didn't want just waste time. Our first assay we evaluated was sarcoma 26 gene panel, and we had research sarcoma group which was highly interested in fusion detection in sarcoma. Genomic translocation represents an important aspect in sarcoma biology, and several gene fusion recurring in specific sarcoma subtypes. Identification of tumor type specific fusion was important for research into targeted therapies. And it was apparent that this assay can be used to identify all of the genes listed on this diagram. And targeted gene here in red could be detected, but also all partners possible with these targeted genes, and also some unknown partners not previously reported also could be targeted. This was very appealing to us. For initial study, we used 35 samples. It was different types of sarcoma, some control lung samples for known fusions, and this basically all samples was positive for some targeted gene by FISH or some other methods. So initially we validated this assay for Ion Torrent sequencing, and sequentially we validated it for Illumina. And the NGS pattern were 100% concordant with available FISH or IHC results, as well as between two sequencing technologies. So some fusion variants were confirmed by RT-PCR followed by Sanger sequencing. So some samples with newly detected stuff, you can have prior results. So we established a confirmation approach. Since Archer analysis allow capture fusion-specific sequence and specific RNA references for fusion genes, we designed primer which flank around breakpoint and perform RT-PCR. So reverse transcription we perform using RNA sample the same sample we used for sequencing with this Archer assay. And I script cDNA synthesis kit from Bio-Rad, and then we follow with PCR fusion specifics primer and run it on 2% agarose gel, which shown here. And PCR product, PCR products, then we run on Sanger sequencing and compare this sequence to sequence presented by Archer analysis. So here you see concordance, the same breakpoint was detected by Archer analysis, by Sanger sequencing. This assay was also useful in some challenging cases we had during our validation, and I want to share two interesting samples. So sample number 1 had a histology of small round cell tumor consistent either with desmoplastic small round cell tumor or Ewing sarcoma. A break-apart FISH demonstrated that EWSR1 rearrangement is present, but EWSR1 rearrangement can have over 40 different partners gene, as it's like shown here from Atlas of Cytogenetics. So EWSR1 rearrangement is common for both DSRCT and Ewing sarcoma, but they have like different partners. So finding right partner here would help distinguish between these two subtypes. And in our Archer assay, we detected that partner was FLY1 and could confirm that this sample is from Ewing sarcoma. Another sample had also histology of Ewing sarcoma, but FISH results for EWSR1 rearrangement was negative. So by this assay, we found different fusion. It was fused ERG, and this rare translocation was reported in only a few cases of Ewing sarcoma. So it also helped to confirm that the samples indeed Ewing sarcoma. We also had some research that we published using this sarcoma assay, and I just want to share a couple of them. First study was detection of the NEP-STAT gene fusion in solitary fibrous tumor. We published it in Journal of Cancer Genetics. NEP2 and STAT6 are adjacent genes on chromosome 12, and they're translated in opposite directions. So due to the proximity of NEP2 and STAT6 genes on this chromosome, this fusion may be missed by FISH. Solitary fibrous tumors is a mesenchymal tumors of fibroblastic origin and can affect any region of the body. And about 15% of SFT metastasize and don't have effective therapy. So behavior of SFTs is difficult to predict based on morphology. And NEP2 STAT6 gene fusion was defined as driving event in SFTs. And from the literature, it was shown that different fusion types could correlate with tumor histology and behavior. We had a small cohort of SFT retrospective samples, which was positive for STAT6 by IHC. And in all of these samples, we detected NEP2 STAT6 gene fusion. And we detected 6 different breakpoints. And as described in literature, despite the small cohort of our samples, we also saw some correlations that shorter isoforms were associated with more aggressive tumors. Another publication was in Journal of Gene Chromosome in Cancer, and it was detection of USP6 fusions in aneurysmal bone cysts. So USP6 is recurrent translocation in primarily aneurysmal bone cysts. And in our analysis, we detected common like CDH11 USP6 fusions, but we also found 7 novel fusion partners. All novel partners we confirmed by RT-PCR and Sanger sequencing, like I described before. So it was quite exciting that we could find so many like novel fusion in this study. So sarcoma panel was very successful for fusion research, and we had a group which was interested in salivary gland tumors. So salivary gland tumors are a heterogeneous group of benign and malignant neoplasms, as it poses significant difficulties to distinguish due to the many different histological subtypes and the rarity of many of these tumors. So there is also morphologic overlap between several subtypes, and molecular studies have uncovered chromosome translocation generation gene fusion network, like shown on this slide, and it's allowed for new molecular classification. So in addition, gene fusions can be investigated for new targeted therapies, and it was appealing for our research group. So based on literature and based on this network I showed you, we designed SGT panel, which included 14 genes targeted for fusion detection. And we also included 2 genes, PRKD1 and HRAS, for point mutation detection. This mutation was associated with salivary gland tumors. We had 62 samples, 60 retrospective specimens, and 2 cell lines in this analytical validation. 49 was FFPE, 6 fine needle aspiration, 5 peripheral blood, and we found that 39 samples was positive, and this fusion was correlated with specific subtype of salivary gland. Here, gene fusion we detected, and like I said, they corresponded type of malignancy. I also want to point out that here we designed this panel in mind to call targeted mutation. So we had PRKD position E710 and HRAS Q61, G12, and G13 position, which was included in design. And we had a list of this targeted mutation in BCF file, which we applied to analysis setup. We didn't aim to detect any of these targets mutations. So compared to sarcoma, we had some interesting new addition to mutation detection. On the next project, we was thinking about lung and thyroid specimens. So molecular testing is important for lung cancer specimens. 3 commonly performed molecular tests in non-small cell lung cancer: EGFR, KRAS mutation testing, and ALK rearrangement. It's usually done by FISH. And then molecular profile of lung cancer is complex and heterogeneous, and molecular testing can provide guidance for therapy. The majority of NSCLC diagnosed at advanced stage and after missing the best time for surgical resection. So the diagnosis and therapeutic decision for lung cancers usually rely on minimally invasive procedures, and we have small biopsies or small cytology specimens for testing. So molecular testing is also important for thyroid cancers, and here knowing point mutation could prevent patients from secondary surgeries. So if you look at NCCN guidelines for non-small cell lung cancers for eligibility for targeted therapy, you will see that require detection of different type of mutations, point mutations, small insertions, small deletions, and also rearrangements in many targeted genes. So we put in our intention to comprehensive thyroid lung cancer, which was off-shelf Archer panel design, and it's include overlapping targets for lung and thyroid. So ALK and TRK red all captured here. Design for this panel included a target for fusions, and also it was expanded designed for CNV and small indels. They also have some genes which they pointed as expression targets. We didn't explore it much. We just started looking into this. So for initial evaluation, we had 8 samples, which was previously positive either by FISH or IHC. And we have also negative and positive control. So results was 100% in agreement with our previous positive finding. And we did additional studies using this assay. So because often we have small specimens and we also have cytology smears, we wanted to apply this assay with different type of specimens. So fine needle aspiration, major first line diagnostic procedure, like I already said, and FNA derived material could be only pathogenic specimens which is available for this testing. We selected best slides for each case, and RNA was extracting using the same RNase FFPE kit which we usually use for tissues. And we obtained enough high quality RNA for library preparation. We didn't have any failures here. And for our studies, we used 27 FNAs, and 11 of them had matching FFPE for lung and thyroid specimens. So these studies was published in journal Cytopathology, and it's highlighted that we can simultaneously detect single nucleotide variants, deletion insertions, and fusion in the small lung and thyroid specimens. So here I want to point out that we did have 3 specimens matched between FNAs, between smears and FFPEs. In all findings, we confirmed by different type of approach, different sequencing. So all RNA detected mutation was confirmed by DNA sequencing, and ALK fusions we detected here was confirmed by FISH, and the same for thyroid specimens. All thyroid specimens had the pair in FFPE. We could detect RET and PPRR mutation fusions, which we confirmed by RT PCR method and single sequencing. And we also found some KRAS BRAF mutation, confirmed this by DNA sequencing. So this is a concordance of FNA and FFP results for fusions in thyroid and lung carcinoma samples. And we also had it on point mutations for FFP and FFPE. This is presentation of BRAF V600E detection. I also want to bring up that this method is reliable for detection of EGFR exon 90 deletion. This activating mutation in non-small cell lung cancer associated with sensitivity to EGFR TKI inhibitors and important to detect, and sometimes they could be challenging to detect by even DNA sequencing. So this method we confirm multiple times is very good for this type of deletions. So this panel was very helpful in lung thyroid research, but we also use this panel on samples for other type of malignancies. So we found 48 positive samples for BRAF fusions. So most often it's seen in brain tumor samples, and we indeed find it that polycystic astrocytoma samples, mostly samples which we found BRAF fusions, but we also have some other malignancies. And here I want to show that we did find multiple partners. So common partners in astrocytoma, but we also found 8 rare and also 3 novel BRAF fusion partners which never was reported in literature before. All of these fusions preserved kinase domain for BRAF and had sense for malignancies we tested. So after using FUSIONPlex for several years, we had demand for additional targets in solid tumors testing, and we also had capacity expand for our sequencing. We had bigger instruments now. We decided to combine all our small panels and expand it to new targets also. So we designed a custom panel using Archer Assay Designer with additional help from IDT Archer bioinformatic team. And it includes 153 gene targets. We also have SNV targets, which for genes shown here in bold. And assay also include 4 control genes for RNA expression. Actually, these genes included in all Archer panels for RNA expression control. So validation samples included 64 previously tested in-house patient specimens. We had some outside specimens, archived specimens from our lab, and also we had 2 positive controls. Majority of samples was FFPE and 5 needle aspiration samples, which we normally see for solid tumors testing. And also we selected different tumor types, with majority being different type of sarcomas, brain tumors, and lung cancers. So we found 46 unique fusions and splice variants in 57 validation samples. So expected gene fusions were identified in each tumor type, and multiple gene targets and oncogenics, other forms such exon skipping in glioblastoma for EGFR, metexon 14 skipping in NSCLC was detected. We also found multiple partners in Ewing sarcoma. We found different breakpoints for BRAF fusions. It was very impressive that we could do everything like in one run, basically, for all this type of tumors and different mutation types. So new finding from analytical validation of this panel. We had to confirm. Here is a liver mass sample tested negative by small panel. During validation, we detected PRCTA fusion, which is consistent with fibrolamellar hepatocellular carcinoma. We designed primers for the specific breakpoint and ran a product on 2% agarose gel, like I described before. And we did forward and reverse sequencing for this sample and got exact match for Archer analysis sequence. Another interesting example of confirmation we had in a breast cancer case. So we had 2 specimens from the same person, which we received over time. One was in 2022 and another in 2024. And one was from brain mass biopsy and another was from chest wall lesion. And we could detect the same mutation in both specimens. One of them with FGFR1 fusion had a non-classical breakpoint in intron. They was identical for both specimens. We also found second fusion in the specimen on both specimens, and both of these fusion rearrangements are associated with invasive breast carcinoma. So it was quite exciting that we could do it in one run. I also want to put here that oncogenic isoforms are important to detect for several malignancies here. So metexon skipping is oncogenic driver in lung cancer and associated with sensitivity to inhibitor treatment. So EGFR exon 27 skipping, most common variant in glioblastoma and associated with aggressive behavior. And another interesting isoform we can detect is kinase domain duplication in EGFR. So it's also potential mechanism of underlying resistance to EGFR. So I want to share a few examples of simultaneous detections of this oncogenic isoform with other findings. So here we had brain mass specimen from glioblastoma, and Archer analysis show exon 1 going to exon 8. So it skipped 27. What is interesting, we also looked at RNA expression tab here. We usually don't analyze expression, but here you can see that EGFR on a high level here. We ran DNA sequencing on this sample, and CNV analysis showing that EGFR have high copy number. So all of this was kind of in agreement, and we could confirm it. Another interesting finding, it's not very often you can detect, and it's quite hard to detect by other method. You can see in Archer analysis, exon 25 going to exon 18 is out of order. And exon 1825 includes the entire EGFR tyrosine kinase domain. And detection of exon out of order could be result of EGFR kinase domain duplication, which has been previously reported in NTRK fusion negative congenital mesoblastic nephroma, which resulted in EGFR activation. For next step, I would like to talk about limit of detection for this method. So we did like kind of good investigation for this big panel because we was concerned about so many targets together. And LOD for FUSIONPlex depend on tumor cell percentage, RNA input and quality, and also RNA transcript expression level. Sometimes it's really hard to normalize all of this in one approach. What we did here, we approached it usual way. We did a mixing study of fusion positive samples with fusion negative RNA, and expected results here shown in dashed line and what we got in reality in blue line. We also had a different type of mutations like fusions and exon skipping in a separate mixing investigation. And sensitivity results demonstrated that gene fusion for this is say around 3% of positive tumor RNA and about 5 start sites. And like I said, the sensitivity influenced by variation in expression level. If you have a high level of expression, you have more start sites at the beginning, and you can detect it to further detection by our method, by our approach. If you have low expression on the first place, it's harder to see it on the lower dilutions. We also investigated a limit of RNA input. We did different input of amount of RNA and prepared library and check can we detect this fusion, expected fusion. And we could detect this fusion in a very low input, but in general, we are trying to have at least 50 nanogram of RNA taken as a safe low limit because you have to have enough fusion molecules for detection. If it's unknown sample with low percent of tumor cells in it, so we wouldn't go for very low input. So we also did a dilution study for single nucleotide variants. So for our analytical validation for large panel, we had 7 SNVs and we chose one BRAF variant, which we diluted with a normal RNA, and came up that 4% of variant reads were reliable limit of detection for this large panel. So all of this finding was confirmed by DNA-based sequencing. It was convenient because we have different type of sequencing in a lab. So this is a positive control we use now with all our FusionPlex. It has 3 different expected fusions. This is commercial control from Horizon. And we also had control with our positive samples mix. So we did reproducibility studies using this control and also FFP samples. So this is a presentation of 8 runs. We didn't have a big diversity in detection of this fusion presentation. This was 100% in agreement in intrassay reproducibility or interassay reproducibility, and we also had 100% concordance with our positive sample previously detected or confirmed, and negative samples were negative. For another interesting panel which we had experience with, it's heme custom heme FUSIONPlex assay. So I will not describe our validation here because we already went through this. It's performed really well in validation. This assay was formulated for detection of fusion in pH-like ALL samples, in also for other heme malignancies, like you can see, we use it for AMLs, for some different type of BLLs. But I want to share one interesting finding here. So this panel could detect IKZF exon skipping. So IKZF1 gene codes transcription factor Icaros, and the expression of this protein is restricted to fetal and adult hematological systems. So isoform leaking, the N-terminal domain can act as dominant negative inhibitor for Icaros, and in B cell acute lymphoblastic leukemia, IKZF1 deletion is associated with a high risk of relapse and worse prognosis. And our research group was interested in detecting these specific isoforms. So this is schematic presentation of this protein, and deletion of exon 4/7 and 2/7 lay in this DNA binding domain area, and we could detect these 2 exon skipping isoform by our heme assay. But what we noticed, we could detect it almost in every sample. We investigated 175 heme specimens, and we found it in over 100 specimens. Then we started paying attention what level of expression of this isoform, and we found that some of them like low level of expression and some of them quite high. We selected 3 groups, low true positive, which had start size under 20 and percent risk between 1 and 10. We had high true positive group where start size had above 10, above 100, and percent risk above 10. And all other, about 79 samples, was expected false positive firing. They had some number of start sites highs that we usually use for positive fusion, but they had very low percent rates. So what we did, we selected a few specimens from these groups, and like I said, this is a start site and percent rates. And we use the approach that we usually use for confirmation. We design specific primers on these isoforms, And took these samples, did RT-PCR with different primers, and ran it on gel and selected true positive, false positive. And as you can see here, deletion isoform molecular weight is different on gel compared to wild type. And like true negative, for example, here showing it's only wild type, so is our suspected false positive here. And this is an example of simultaneous detection of these isoforms. This is like two isoforms in one sample, and also BCR able in the same sequencing run. And another example in this sample, we observe a simultaneous detection of CRLF2 fusion and IKZF1 exon 27 skipping, which might be important for prognosis. And another useful approach with this panel is detection of resistance mutation. So ABL1 can ABL1 mutation T315 is the most common resistant mutation, and because of this, we did serial dilution for this mutation, and we started with very low variant percent reads. We had samples with 3.4% reads, and we determined that we can see this mutation down to 0.5% of mutated reads. So also you can detect that mutation by this method simultaneously with your fusion detection in one sample. It was also kind of important, and we see it not very often, but sometimes it could explain why these tumors behave this way. Heme panel is quite popular among our research groups, but it has some limitations. Only rearrangement resulting in fusion transcript, not involving with IgH, are detectable by this panel. So rearrangements with IgH alters the expression of the partner gene, but does not alter the gene transcript. Several disease-defining rearrangement may be missed, and it's requiring traditional assay, and is it there or not? So pH-like ALL tumors with CRLF2 IgH or 4 IgH fusions will be missing by this assay. It's need alternative testing. So because of this, for our future directions, we would like to address IgH rearrangements, and we know a couple groups try to do it based on this heme arrangement using partner gene expression level. For our future directions, we also would like to expand our heme panel. We know that Archer have a large 199 gene panel, but we also had some requests for additional genes. We already looked in this panel, and I think it's our next step in this direction. And we also would like to go for library preparation automation. Archer expanded to liquid reagents, and lately we have increased in our research samples. So it's take quite a bit to prepare this library now. And with this, I would share our team with you, these people who participated in this research, this part of this like clinical research teaching group, and I would like to thank all of them. And here is University of Iowa Hospital and Clinic in the mid of United States. And with this, I would like to thank you for your attention, and would like to answer your questions. I would like to share what we have in our use. Thank you.
Thank you so much for the wonderful presentation. It was really great and very informative. Now we can jump into Q&A. So let us kick off with the first question. What do you use for reporting and variant interpretation?
So generally, like I showed on my slides, our analysis going for Archer analysis provided software. We have it on virtual machine here. We don't go in clouds, but for interpretation and finding and QC, we rely on Archer analysis. And it was sufficient for us so far. Archer analysis allow to detect a special breakpoint for fusions. It already did all annotation, all secondary analysis going there. We apply our FSQ file, and Archer analysis do this all background for alignment, detection of this REF6, and it's also provide visualization for decision making, like calling mutations. We can decide how fusion look in browser, and in addition, if you have higher, for example, new partners, we would search literature. So Archer analysis also provide the database keyword they curated, and if it's found in this database, you will see it flagged. Otherwise, you will have to do PCR confirmation and literature search. And also CNV, we rely on Archer, but since we are aiming for specific mutations with our specific file, we don't aim to find somewhere in novel mutations for annotation. It's quite easy.
Thank you. So for the next question is, would you want to perform QRT-PCR for fusion validation?
For fusion validation, we do use RT-PCR from the same sample if we have sample available and if we have novel mutation. For common mutations listed in database, usually we are very familiar with this technology. It's we don't have any doubts. This is like validated through positives. But if you have novel fusion or any doubt in breakpoint, for example, we are going to perform RT-PCR and we are going to perform single sequencing and compare it to sequence from Archer. About qPCR, I'm not like sure. Is it about quantification? But we like for confirmation, we do RT-PCR in a Sanger.
Thank you. So then for the next question is, what challenges do you face when evaluating the reliability of a detected fusion?
So I think most challenging with assay, and we was quite successful with this, is nature of our samples. If you have really poor quality RNA on the first place, everyone knows it's really hard to work with FFP and RNA, especially if you have very old samples for research. And in this case, like you have to decide at the end what you can do. And sometimes increasing input could help resolve these problems. You will see more molecules for initial analysis. But generally speaking, fusion detection was quite robust for so many years. I couldn't tell you like on discrepant call where we had doubt. Like generally, if it's something in question, it's sample issue. And we also have some outside testing for our samples where we could see, oh, we found it and it wasn't found before. In many cases, our testing for our research was true positive.
Great, thank you. So do you use any of the Archer product DNA panels? Do you see added value for using both?
This is an interesting question. So we started with DNA sequencing on different approaches. We started point mutations and expanded it to panels. We have multiple approaches how we do it in the lab. So for us, most important was fusions. We do run the same sample in parallel. We extract it as TNA. We use RNA for Archer analysis, and DNA we sequence with different platforms. VariantPlex, we had a limited experience, and for us, it didn't make sense because validation of pipelines was very intensive. We already have validated approach for DNA calls.
Thank you. So, have you ever seen EWSR1 cases that are FISH positive but FusionPlex negative?
I showed you a couple examples. Usually, we find that FusionPlex more sensitive than FISH. FISH sometimes hard to interpret, and sometimes FISH can detect fusions relative to these malignancies. Plus, even if you see partner EWSR1, you have to know another partner, right? Yes, we did have cases which was positive by FusionPlex and negative by FISH.
Thank you. So, for the next question is, how often do you see false positive fusions, and how do you determine that they are false positive?
Uh, Again, not very often. Usually, when we have a number of start sites above 5 and number of reads above 8, we are pretty confident that this is a true positive call. So we had it with like very long experiments in validation of these panels. And sometimes you see this presentation, we usually compare it to our tumor content, and you can see, oh, it's very low, but number of tumor cells was also low. You wouldn't normally test it for clinical, for example, because it's below 10%, for example. But FusionPlex is positive, and I think I'm pretty sure about this technology. There's known fusions which on a very low level, and they would go above 5 start sites. They're true positive. Uh, Saying this, like using this big panel now, we find very interesting weird stuff sometimes, which have like targeted gene which related to malignancy we tested for, but on another side, it's very unknown partner. For example, non-coding RNAs, and it's not very well known, and we have to confirm them. Of course, they usually confirm by RT-PCR and Sanger, and then we have to make sense out of this. What does this really mean? And some literature on this exists, and it's saying that it could play a role in development of these malignancies. And we are working on investigating this in our next research studies.
Thank you so much. So, given that we're approaching the top of the hour, that will be our last question for today. But I just want to say that on behalf of the entire team, we want to extend our sincere thanks to everyone who attended today's webinar. A special thank you to our speaker, Dr. Guseva, for sharing such valuable insights, and to Am for hosting the session. We truly appreciate the thoughtful questions and engagement from our audience. If we didn't get to your question during the session, don't worry, we'll be reaching out directly with responses. So thank you again for your time and participation, and we really look forward to connecting with you at future events.